¿Me escucháis?
Right, can you hear me? First of all, I'd like to thank Carmen for her invitation and for having me here again to tell you how the gene therapy is developing. Ida as well for the organization, for being so proactive, so reactive all day. I don't know when do you sleep, and the foundation Per Amor al Art, the association Faustino, for everything, for gathering us here. I've been coming for a few years, and I think the first time there was only 20 attendees, so I'm so happy to see a full house and in such a wonderful place. I think many of you will see some of the slides that I have presented before, but we have new faces, and we don't talk about gene therapy every day. So I'll go back to basics to tell you what a virus is, how we modify them, so you can understand some of the results that I'll be presenting. I will not deep dive too much, because I'd like to cover the latest data on gene therapy, because right now there's been a huge quality development at present. Right, my disclosures. I work in the University of Navarra. I'm a researcher, but I'm also a co-founder and chief scientific officer of Vivet Therapeutics. So this is the summary of the overview of gene therapy, what is the adeno-associated viruses, and where have we used these AAVs, and with all these in the treatment of Wilson's disease, and starting with the complete ATP7B gene, the mini genes, the 2 AAV vectors to cure the disease, and finally the gene integration, the gene editing, which is probably the future to the treatment of genetic-based diseases. So gene therapy, well, this is very easy. It's basically using genes to cure a disease, and that's all. With monogenetic diseases, when we know that which one is the target organ that we have to cure, the liver in this case, what we know is that we need to correct the gene, the mutated gene, to the cells where the disease is produced. In our case, the liver, where is the complex part, how we can make these corrected genes to get into the cells. It's not easy to introduce the gene material into our cells, but basically there are some viruses that can do this very efficiently. Viruses basically only do that. They introduce their gene material into our cells to produce their own proteins and be able to replicate and sort of move from one person to the next. What we do is, using that property of viruses, we modify them genetically. So instead of introducing their gene material, they introduce what we want them to. What viruses are we going to use? So you'll be able to see very attractive viruses to be used in the treatments. These are the adeno-associated viruses, AAVs, very tiny viruses, and they're called AAV because they were adenovirus contaminants. The contaminants of adenovirus, they produce upper airways infections for children. Basically, all these infections by children are produced by adenoviruses, and this is how they appeared. They were contaminants of these viruses, but they have very interesting properties. Naturally, they do not replicate. So you have an adeno-associated virus. The virus penetrates your cells, but it doesn't create new viruses because it's missing important proteins that some other viruses give them. such as adenoviruses. So these are tiny viruses, one of the smallest viruses that exist. And they have only one protein. This is the one that forms this capsule. It has this little ball with the protein, and inside it has the genetic material. And there's another property that makes it very attractive. It doesn't produce any disease. All of us, we've been infected by adeno-associated viruses, and we don't know, because we've never developed any symptomatology, either fever or anything, because we cannot say I've been infected by an AAV, because you don't know. So from the therapeutical viewpoint, this is very attractive. This is the AAV inside. This is what we can see at its core. This is a DNA sequence, and such as the one we all have, but these viruses only have this tiny sort of stream of DNA with two genes. The rep gene, it encodes all virus proteins involved in replication, and the cap gene, which encodes all virus proteins involved in virus encapsulation. And at its ends, basically, it has some sequences called ITRs that look like hairpins, and they indicate that this is a virus. So when you have this sequence at its ends and a longitude of 4.7 kbs, this is identified the DNA of an AAV. So all these viruses that we produce to cure, basically we get rid of all this genetic material in the virus, and we only leave the ends. So the cell is misled and believes that that's a virus, and then it can create new viruses from that particular material, genetic material that we're going to introduce, and we'll see that in a minute. As I was saying, what we do is we get rid of the replicat, which are the rep and cap, and we substitute it with the therapeutical genetic material. And what is that? The ATP7B gene and other DNA sequences that I need to introduce so that the protein is expressed where I want it to be, and it will be some gene material that will make this protein to be expressed on the liver cells. So what happens when we do this? As I was saying, we need to keep this these ITRs at the ends, but when we get rid of the rep gene, which makes the virus, when it's introduced in the cell, remains within the chromosomes, when I get rid of that, that virus doesn't integrate into the cell chromosomes. It stays outside of the chromosomes. What does it mean? That when the cell is divided, the virus genome is lost. That is important, and you'll see that very soon, because in a growing liver, the liver cells split, and the gene material of the virus will disappear, and that might be a problem if we have to treat children where the liver needs to grow, and it needs to these cells still need to divide or split up. There's another limitation of these AAVs. They are tiny, as I was saying, and we can only introduce a maximum of 4.7 kilobases, and this poses a problem for Wilson's disease that we've managed to solve, as I'll show you. So when I want to produce a recombinant AAV, the only thing I have to do is to introduce and collect all these necessary elements, the recombinant genome with the therapeutic gene, the rep and cap proteins, and the proteins from the virus that help, like the adenovirus proteins, and all that, basically I do a cocktail and I introduce that in a cell that will be able to produce viruses that I can purify and will be used to treat the animal models first and then human patients. These are some of the studies that have been published so far using AAVs in the treatment of Wilson's disease, and I'll be trying to summarize in a simple way. This is the first sort of work that we did with Wilson's disease and AAVs, work that we initiated thanks to the Peramoral Art Foundation. And what we did was we used the gene ATP7B. We introduced the gene, complete gene, in an AAV under a sequence that would allow its expression only in liver cells. This example, you can see that this is a mice with the Wilson's disease treated with this AAV, and how we can get rid of copper of that liver. This is a mice with Wilson's disease with a lot of copper in its liver, and this is after the treatment. So these are the results that we obtained in this first study. As we can see, the treatment with this vector expressing the ATP7B protein managed to deplete the urinary copper levels in a normal, healthy mice. This is a mice with a Wilson's disease, and the treatment with the vector with two different sort of versions would allow us to normalize the urinary copper levels, and it would allow us to normalize the transaminase levels in all the different times where we assessed it with the tests. 24 weeks later on, the transaminase levels were under control, and when we assess the ceruloplasmin levels at a high dose of vector, we would manage very similar levels to compared to a healthy mice, and we would also deplete the copper levels close or very similar to a healthy mice. Something important here to mention is that this would take place with the Wilson disease male mice. With male mice, we could sort of normalize all those parameters that were impaired in a model or in Wilson's disease, and we would do that in a stable way. However, when we were treating females, we started to see that we needed to administer higher doses of the virus used with male sort of mice. Even the urinary copper levels were maintained normal, the transaminases were under control. We would need to reach higher doses of the vector to try to get the ceruloplasmin higher, increased, and even though we would decrease the copper levels in the liver significantly, we wouldn't be able to normalize them in females.
Pues igual, subiendo simplemente la dosis.
We increase the dosage in the vector, we will be able to obtain a better result. But what did we realize is that this virus was larger than the optimal capacity that AAVs have. So when we were producing, we faced issues when we wanted to have a higher titer. Therefore, we decided that instead of working with the large version, the big version that was presenting issues, and in order to take that to a clinical setting was going to be quite a challenge, because mice are 20 grams. A human patient is a bit heavier than 20 grams. So we will have an issue if we really wanted to get to the patients, because at the end of the day, that was our objective. So we were working of re-engineering the protein, reducing the size of the gene so we could introduce it really in the AAV genome and for it to work. and we were able to develop a version by which we actually eliminated 4 links in the ATP 7B, and we were able to reduce space, and then we will be able to have genome that will be optimal. This is our mini gene, and then Ultragenyx developed another mini gene where it was slightly bit larger than ours, and they were introducing here a link to metals 4 right there. You can see vis-a-vis the Vivet versus the Ultragenyx, but the efficacy, therapeutic efficacy, is working in both male and female mice, and we were able to see that at least 1 year of treatment, we had urinary copper levels at the levels we wanted. These are WD mice, these are healthy mice, and really there's no difference of those treated with our vector. We increase survival about a week 40-44. Those presented with WD started to die, but those that had been treated with our vector were totally healthy. And the same happened with the levels of transaminase. So on those mice that were treated with our vector remained without any hepatic damage. And after a year of treatment, and we've been treating copper levels, and the copper levels are normal, are practically unnoticeable vis-a-vis healthy mice. On the other hand, and just like Hans presented, we wanted to know if our vector was actually recovering the physiological metabolism of COPA. Unlike the chelating agents that eliminate via urine, we wanted to see, so we actually used radioactive COPA and see, and this was the Aarhus in Denmark actually validated this through the injection of copper in the liver. We saw that prior. And these are healthy mice after radioactive copper, very low levels in the liver, and here we detect that in the gut, bonded to feces, and this is WD mice, and everything is retained at the liver. But on those that we treated with our vector, with VTX801, as we increase the dosage of copper, they start decreasing at the level, at the liver, and they start appearing at the gut. So we are capable of expressing the protein that we need at the liver, and that such protein can do it and eliminate and do the elimination via the feces, first gut and then the feces. And we see radioactive increasing as we increase the vector, and to the highest dosage, there's no difference vis-a-vis or versus a regular mouse. And the one that was done with Ultragenyx, where they used the 4, it's just a slightly different version of this mini gene. They were also able to see how this vector treatment is able to reduce liver copper and also increase ceruloplasmin activity. Both of vectors that I've told you and describe are at the clinical trials. I remember at the beginning of these gatherings and sessions where we all said, oh, gene therapy is not going to get here on time. It has taken time, yes, and it has been painful. It hasn't been a rose rosy path because it requires a lot of testing and a lot of failures. But at the end of the day, we need to be hopeful. We are now with clinical trials, both ourselves and phase 1, phase 2 Ultragenyx. So we're moving forward. And now I want to share what I think is the future, what we hope will arrive soon for this. Maybe not the best, not the first technology, but when I mentioned the second technology, definitely that's the future. Here, this is another strategy that we have, which is genic addition. ATP7B is quite large, and it really imposes challenges that at the beginning were quite hard to bypass when we started. But now there's a group, what they have done is they have a split ATP7 gene, so the protein has two different segments, and they have actually used a bacteria. So ATP-B7, the protein is then joined, and they, at the end, it's always, should we eliminate these pieces of the protein? Are they going to affect or not? How it works? They work in Naples, and this is what they have done. Use two AAVs. So these two pieces or splits of the ATP-B7 and then put them together again. And what we see, the results that they have published, what they have noticed is when they treat WD mice with these two vectors, they are able to decrease the fibrosis level of mice if compare non-treated mice. Pathology at the liver is better, but they're not able to decrease hepatic cover. Here we have green sick mice and red and kind of brownish. If you see it on the screen, they're treated with with these two vectors. So even though they are not reducing kappa, they are able to improve the histology, hepatic histology, which is also important from the disease point of view. They're able to reduce significantly hepatic kappa and brain kappa, and the expression of kappa through excretion of kappa through urine. Now, this second strategy, this is quite the future, is to integrate this gene ATP-B7B. So they use this mini gene and they eliminate 4 metal spots, and they are now forcing this mini gene at the site where we find the albumin gene. The albumin gene, albumin is a protein that is expressed at very high levels at the liver. This group, they take advantage of that in order to introduce this mini gene, ATP7B, so it is under the control of the sequences that regulate the expression of albumin at hepatic level. This strategy that actually favors growth, because the corrected cells are going to have an advantage. Those non-corrected cells are going to, we're going to see apoptosis because of the accumulation of COPA, but those that are being helped, they will have a selective advantage compared to those one in a mice, where you see the liver is growing and the hepatocytes are growing and increasing. This will allow for the new hepatocytes, for this new liver cells, to have the gene incorporated in their own chromosomes. And as a matter of fact, I know you cannot see it well on the screen, unfortunately, but if you take a look here, this is a mouse that received this vector, and the gene, the mini gene, has been able to integrate in the hepatocytes, in the liver cells, and is repopulating the liver of this mouse. So when we treat mice at 6 weeks, the liver no longer is developing or growing. Young individuals, 6-week mice, they improve the status of the liver, but they don't really reduce copper, which is similar to the first new technology that I showed you just now. So this is related to the fact that when you reduce the transaminase levels, but they don't get them to normal. Even though there's a reduction, they don't go to normal. Something different is when the mice are treated at the age of 2 weeks. This is a mouse, or their mice. This is it would be a young child, if you compare it because of the age, 2-week mouse. And then you see here the copper. They're able to reduce copper in the liver. So this is a strategy that that is going to be quite attractive for treating pediatric patients, and otherwise this will be diluted in time. And this is my last PowerPoint, so I just wanted to say thank you. Thank you all for being here, for being supportive, and thank you to the sponsors, to the grantors. Thank you to Fundación per Amor al Arte since the start of all times. Thank you all. Thank you.